Myself, Professor Sujata Mahapatra from the Department of Botany, Ramadevi Women's University. In this lecture, I shall be discussing about yeast to hybrid screening or yeast to hybrid assay system. Yeast to hybrid assay is a in vivo method for the study of protein-protein and protein-DNA interaction. This method was invented in the year 1989 by Fields and Q. Song. The importance is that it can be used for the screening of the proteome. An entire protein library can be screened by this particular process. This is a genetically engineered assay system, and the process involves the transformation of the yeast cells with genetically engineered plasmids. The haploid yeast cells are cultured on the culture plates. Then diploid yeast cells are selected and they are studied for protein-protein interaction. There are certain requirements for this type of assay. Mutant yeast cells, Saccharomyces cerevisiae, mutants which are lacking certain biosynthetic pathways are selected for this purpose. Then a selective medium for the culture and selection of the diploids is required. Two types of genetically engineered plasmids are used. One plasmid construct, it carries the gene for the DNA binding domain of the transcription factor and the gene that produces the bait protein. The second plasmid construct carries genes which code for the activating domain of the transcription factor and the genes that produce the prey protein. These proteins, bait BD protein and prey AD protein, are produced as fusion proteins. A reporter gene is needed. There may be lethal genes, antibiotic resistance genes, or genes for the biosynthetic pathways as discussed in this slide. The yeast to hybrid assay system is based on the principle that each transcription factor, not only in yeast, but any transcription factor, has two domains. One is DNA binding domain, BD, and the other is the activating domain, AT. And these two domains are structurally and functionally independent. So even if BD and AD are indirectly connected, they can reconstitute a functional transcription factor. BD binds to the upstream activating sequence and AD is required for transcription. And in the process, as we'll see later, how it is made, the yeast gene coding for the BD is ligated to genes coding for the protein of interest of the bait. This gene construct produces a fusion protein with BD of the transcription factor and the bait. Bait protein binds to the N-terminal domain of the BD of the transcription factor. In the second gene construct, the yeast gene coding for AD is ligated to the genes for proteins of prey to prepare multiple gene constructs. The prey proteins are picked from a library of proteins, and the genes for those known proteins are used to prepare the second gene construct. And this gene construct produces a fusion protein with AD domain of the transcription factor, and the prey protein binds to the C-terminal domain of the AD. So now we have two different plasmid constructs, and these two plasmid constructs are introduced into separate yeast cells. After that, transformed yeast cells are isolated and allowed to mate. The next step is isolation of the diploid yeast cells and screening for the expression of the reporter gene. Yeast 2 hybrid system makes use of the modular nature of the transcription factor. Modular means the transcription factor can be separated into independent components which can interact and which can reconstitute the whole factor whenever the situation arises. So in this assay system, the transcription factor is active if DB and AD are connected indirectly too. So they are independent, but they will be functional when the DB interacts with AD, but direct interaction is not necessary. Now the host cell, it expresses the reporter gene under certain circumstances when the DB and AD are indirectly connected through the binding of the bait and the prey protein. When the bait and prey protein are compatible, they will indirectly join DB and AD of the transcription factor and reconstitute the whole transcription factor that will help in the expression of the reporter gene, which produces an observable product. This diagram shows how the transcription factor acts. The transcription factor has two domains, DNA binding indicated by red and the activator domain indicated by purple. They recruit other transcription factors and RNA polymerase becomes functional and reporter gene is expressed. So here in the next picture, the DNA binding domain and the activation domain are separated. The DNA binding domain is linked to the bait protein and the activator domain is linked to the prey protein. If there is compatibility of the bait protein and the prey protein, then they will interact and reporter gene will be expressed and a squarable product will be produced. This is how it is shown here. There is proper interaction. Here, there is no interaction. There is no compatibility between the bait and the prey protein. That is why, though there is no joining of the transcription factor, there is no reconstitution of the transcription factor, and reporter gene cannot be expressed. But in this diagram, there is compatibility. There is proper interaction between the bait protein and the prey protein. Two components of the transcription factor, the BD and the AD, are indirectly connected by the interaction of the bait and the prey protein. Then RNA polymerase is activated and reporter gene is expressed. For yeast 2 hybrid assay, two types of plasmids are engineered. One is bait plasmid, the other is prey plasmid. Both the plasmid have the origin of replication and the marker gene. These are not shown in this diagram. The bait plasmid has the genes expressing the DNA binding domain of the transcription factor and the gene expressing the bait protein. The prey plasmid carries the gene expressing the AD domain of the transcription factor and the gene expressing the prey protein. So these two plasmids are constructed to transform the mutant yeast cells. Haploid yeast cells are used for transformation. One haploid cell carries the plasmid that will produce the bait as the fusion protein in addition to the DNA binding domain of the transcription factor. The second haploid yeast cell will produce the prey protein, which is also produced as a fusion protein with the AD of the transcription factor. Both the haploid yeast cells are histidine negative. They cannot grow in a medium deficient of histidine. If the mating is successful, then a diploid yeast cell is produced. It may be negative, it may be histidine positive, depending upon the interaction of the AD and the BD through bait-prey interaction. So we can see two plasmid constructs in this particular diploid yeast cell. In the next step, the diploid yeast cells are cultured in a medium deficient of histidine. If the proteins interact, diploid yeast cell will grow because the reporter gene will be expressed and histidine will be produced by the diploid yeast cell that do not require histidine from the external source. That is why they will grow in a medium which does not contain histidine. This is a haploid yeast cell which is histidine negative. This is the second haploid yeast cell, also histidine negative. This haploid yeast cell carries the gene that will express the DNA binding domain and the bait protein. This will express the AD of the transcription factor and the prey protein. After successful mating, a diploid will be produced, and in this diagram, it is shown that the AD and the DB are interact because the bait protein and the prey protein interact. So the two components of the transcription factor, DB and AD, are connected indirectly through the interaction of the bait and the prey protein. So the reporter gene will be expressed and this diploid yeast cell will grow in a medium that does not have histidine because the diploid yeast cell will itself produce histidine because of this reporter gene. This is the gene for histidine, histidine 3 gene. So histidine will be produced by the diploid yeast cell and it will survive in a medium that does not contain histidine. If the bait and the prey protein do not interact, then they will not connect the DB and the AD of the transcription factor. So transcription factor cannot be reconstituted and histidine gene will not be expressed. That is why this diploid cell cannot grow in a medium that does not contain histidine. No histidine from the external medium and no histidine by the diploid. That is why it cannot grow, it will not survive in a histidine deficient medium. This is a complete picture The bait has DNA binding domain and the target protein, and the prey is the transcriptional activation domain and the binding partner, any protein of interest. They are introduced, and if interaction is there between the bait and the prey, then transcription of the reporter gene occurs and reporter protein is made. In the library screening approach to carry out the yeast to hybrids assay, the liquid culture of two types of yeast strains are prepared. One liquid culture is the bait culture, the other is the prey culture. The bait culture has yeast strains, haploid, and which produce the DB and the bait protein, fusion protein. And the second set of culture, the prey culture, has haploid yeast strains which produce prey and the AD as the fusion protein. Inoculum from both the yeast strains are picked up and allowed to co-culture on a nutrient-rich medium. Next step is the isolation of the diploids. So the next, again, diploids or the cells which grow on this medium are picked up and grown on a selective medium, then on multiculture plates. The selective medium contains IPTG or it may be without histidine, and if the bait and the prey interact, then only the diploid will grow in a medium that does not contain histidine. Then for multiplication of these yeast cells, they are cultured on multiculture plate till further analysis, and subcultures are made from time to time. In the matrix-based approach, a liquid culture of the haploid yeasts which express the bait protein is prepared. The prey protein expressing yeast cells, which are of the opposite mating type and haploid, are spotted by robotic printing technique on a 96-well plate. This is a microarray plate. Each well contains one haploid yeast cell which produce the prey protein. In the next step, one haploid yeast cell is picked up from the bait culture and added to one prey producing haploid yeast cell, so that each well contains two haploid yeast cells of the opposite mating type. They are allowed to mate in a nutrient-enriched medium because each well contains a nutrient-enriched medium. After the scheduled time period, from these wells, possible diploids are picked up and they are cultured on a selective medium. Some wells may not have diploids. Some wells may have diploids without any interaction of the bait and the prey protein. All are picked up and cultured on a selective medium. In the selective medium, only the yeast cells which are diploid and in which there is interaction of the bait and the prey protein will grow, because these diploid yeast cells, after interaction of the bait and the prey protein, will reconstitute the transcription factor for the reporter gene, and the reporter gene will be expressed and it will produce a protein that will enable the yeast cells to grow in the selected medium. So the yeast cells which are growing in the selective medium are the diploid yeast cells, and in which the bait and the prey protein interact. So these are the diploid yeast cells, and in which there is a interaction of the bait and prey protein, and these are preserved by replica plating for further analysis. There are several applications of yeast to hybrid system. It is used for the analysis of different interacting proteins which participate in cell cycle, in programmed cell death. It is used for the study of biological network, that is interactome analysis, which studies protein-protein, protein-DNA interaction. It is used for the analysis of interaction between viral and human proteins, for the distribution and functional study of the cellular components, study of host-parasite interaction, and in plants, it has been used for the study of post-translational modification of some proteins in case of Arabidopsis. Yeast to hybrid system is scalable. Scalable means it can be performed in a small scale, or it can be used for a high-scale protein interaction study or high throughput technique. Living yeast cells are used, it is in vivo technique. So any interaction can be correlated to the actual interaction taking place inside the living cells. As a model system, it is very good and it is very convenient model system for the study of protein-protein interaction. It is a very cheap method and rapid assay technique. As with any other technique, this technique has its limitations. Mostly, there is a problem of false positive and false negative. So, it is not the final interaction study. Yeast to hybrid system can be analyzed further by using other techniques. Thank you.